few minutes. So I'm going to do the introduction today for Grand Rounds. Our speaker today is Dr. Chad Petit, currently an associate professor here at UAB in the Department of Biochemistry and Molecular Genetics. He's been at UAB for, I guess, almost 10 years, since 2012. Came to UAB after completing his Ph.D. in molecular virology at LSU in Baton Rouge. After that, he completed a postdoctoral fellowship in structural biology and biophysics at the University of North Carolina. His research area now focuses on host pathogen interactions involving both influenza and coronaviruses. He's really known for his expertise in NMR spectroscopy and uses that to study how viruses proliferate, which of course is very important in the development of medications, antivirals. He's co-director of the Biochemistry and Structural Biology graduate training theme for the department, also an associate scientist of cancer cell biology for the O'Neill Comprehensive Cancer Center, and also an associate scientist for the CCTS. He's going to talk to us today on structural insight into modulation of influenza, non-structural protein 1 intracellular localization. And so we'll look forward to that. Thank you.
Thank you for the introduction and the invitation to talk every day. I mean, talk to you guys and gals today. I don't mind if anybody has any questions, please interrupt.
Variance sigma squared. I'm going to make this transformation. I'm going to write it the same way that's in the slides. Now I'm going to.
Young son, turn your mic off.
Times y minus mu over sigma.
I'll see if I can keep going.
So cut up the numerator.
You may be able to mute him.
Okay.
Daniel, you might be able to also.
This new random variable is a standard normal random variable. So if I take a normal random variable, subtract its mean, divide by standard deviation, that new transformed variable is a standard.
There we go. Okay. Sorry. As is everybody. Now my computer is frozen. Okay. So as normal with Zoom, I'm much more comfortable giving talks in person. So, you know, I'm kind of just sitting here in like a radio booth talking. And so if anybody has any questions, please unmute yourself and I can explain things further. I don't mind, you know, I like, you know, sort of a conversational style of presentation. So please just jump in. So like Daniel said, I'm going to be talking about structural insight into modulation of influenza non-structural protein 1. Okay, so to just basically get started, you know, we first have the influenza variant particle. It's a member of the orthomyxoviridae of viruses. It's negative sense strand, a single strand RNA genome, and it's about 80 to 100 nanometers in size, and the genome's segmented, which is very important for one of the aspects of influenza pathogenicity and pandemics and things like that, and which I'll kind of be going into a little bit in a couple of slides. So it's 8 segments, but it's, you know, it codes 11 proteins because of different reading frames and frame slippage and things like that. And interesting, you know, trying to get into the nomenclature of influenza viruses. So like unlike the current COVID pandemic, where you have like these alpha and beta and gamma and delta, obviously that's important, strains or variants. In flu, it's sort of a little bit more detailed. So for example, like this is Spanish influenza, which unfortunately I used to say was the most deadly pandemic in history, but I think we're gonna pass that with COVID any day now. So the subtype is H1N1, and that's based on the antigenic responses for the surface glycoproteins. And so, for example, if you have 1918 H1N1 strain or Spanish influenza, you can have the virus type. There's influenza A, B, C, and D, actually. You have a geographic origin. It's a breving mission. It was a mission in Alaska where they found this. Actually, pretty interesting story is they actually dug into the permafrost and excavated some of the bodies that died. And they're able to get the sequence from the people that had died, and they were preserved in the permafrost. So that's how we know the sequence of Spanish influenza. you have a strain number here. And since, I think there might be a couple more now, but since that was the only body that they had, or cadaver that they had exhumed, they only had one, the year, and the subtype. And so if you hear like H1N1 or H7 or H5 and I forgot what the high path virus is, but frequently you'll say like swine flu, H1N1, and that's sort of how they're known colloquially. And so that's whenever you hear that on the news, that's what that means. So why should we, the taxpayers of America, care about influenza? Okay, so every year you have unique strains of influenza that cause seasonal outbreaks, usually about 200,000 hospitalizations. The economic burden due to such things as not being able to go to work and things like that is around 87 billion. And the annual impact is largely determined by each strain's ability to effectively be transmitted from one host to another, or transmissibility, and the severity of the disease state caused by the virus or virulence. And so it's kind of interesting in the times that we're living in that a lot of these terms, while I used to have to explain them to non-virologists and particularly my family, have become sort of common parlance in our times. Just people sort of talk about this much more. It's very surreal in some aspects to have your job basically become matter of public, unfortunately, public concern. So one of the consequences of a segmented genome is that, say you have a highly pathogenic avian strain and you have a human strain that affect the same host. Now, if they affect the same cell, there's nothing really that stops any of these segments from joining together and creating some sort of newly highly pathogenic human strain. This is referred to as reassortment. And this has occurred, throughout history. I think there have been 5 or 6 recorded pandemics for flu. And typically, this is really bad because, say you have this highly pathogenic avian strain. There's also swine flu strains, and they're really pathogenic. I know that H7N9 right now is about 80% lethal. It's an avian virus, but it doesn't spread. So the nightmare scenario is if you sort of combine the human spreadability and be able to sort of have the correct receptors for the human cells with the lethality of the avian strain, and you have a highly lethal strain that can be passed human to human very readily. That's sort of the nightmare scenario. And another thing is flu affects a lot of different types of animals. For example, there's, obviously avian species, swine, even whales. They've actually, sorry, let me turn this off. Actually, we're able to find an instance where that whale had the flu, and they didn't know how it got it, but one of the aspects was you had a seagull or something that had influenza, and the droppings went into the sort of blowhole at the top, and that's how you contaminated or infected a whale with influenza. I thought that was one of the most interesting cases I've heard for flu. But it basically has unlimited animal reservoirs. It's highly mutagenic. You can have reassortment, and that's why there's sort of, when I started studying flu, it's flu's here to stay. It's endemic. It's not going to go anywhere. So the 19 influenza outbreak was so bad that it actually decreased the life expectancy in the United States. You can see this is a pretty horrific graph of the average life expectancy, and suddenly, for a year or two, you had this dramatic drop off, and that's due to the influenza pandemic, which was happening around World War II. So the idea is that, was it that much highly transmissible, or was it that these different military units were going around and spreading it unnaturally throughout the world? We really don't know what exactly caused such a high fatality rate, but it was pretty up there. And so this sort of, when I was thinking about my research program, this really led me to the question, like, okay, so in 2009 we had an influenza pandemic. This was known as the swine flu. And so you had, 3.03% case fatality rate, about 18,000 deaths, whereas the 1918 strain had about 20 to 100 million deaths and 2% fatality rate. And so, you can conclude that even if it was able to spread from patient to patient or human to human very effectively to cause a pandemic, it was substantially less virulent. And so that's what I started, sort of framing my research around why. And so, what proteins would likely to contribute to virulence? And so you have the hemagglutinin that's responsible for entry, matrix 2, which is a proton pump that allows the virus life cycle to continue. You have the neuraminidase, This sort of catalyzes release. Matrix 1 forms the actual virus particle itself. Nucleocapsid protein, which interacts with the vRNA. So, you have entry, release. So this is probably spread. You have, this keeps, allows the virus to keep going because you have a lower pH, which causes a structural change in hemagglutinin, which then allows the virus to, sort of merge and release the genetic material. Plus you have two that are structurally, in the protein, the variant itself. And so far, okay. So then we had the polymerase, which is heterotrimeric. It's PA, PB1, and PB2. They have had instances in mutations that increase virulence. But this might be sort of a sidestep in that if you have a polymerase complex that is much more efficient, you might produce more viruses, which then in turn make a much more virulent or pathogenic strain. Non-structural protein 2, it mediates the export of the RNP or ribonuclear protein from the nucleus. And then we can then we come to non-structural protein 1. Let's see. All right. And this inhibits cellular defenses against viral infection, and it interferes with the cellular processes allowing viral propagation. So essentially, it inhibits the innate immune response. And the innate immune response is very important, obviously, to viruses because that's sort of the first line of defense. So why should we care about NS1? Well, the current treatment options, we have, it's actually just 3 classes. I updated this slide recently because there's a new class. Typically, they targeted neuraminidase, matrix 2, which is the proton pump, and the polymerase. And so although these effective treatments are popular, the emergent circulating strains resistant to both classes are all 3 classes. I'm not sure about the polymerase, but at least for these 2 classes have been found. And so one of the particular antivirals was for the matrix 2. It basically was started to get the medical community started treating it like a prophylactic to prevent flu. And within 2 years, we went from having 2% of the viruses resistant to animetidine, which was the actual compound, to 98%. And so we don't even use that anymore because it's just, we basically created a selection pressure to where the virus was able to mutate away and is now highly resistant. And even in 2009, there's 98% of the seasonal strains were resistant to oseltamivir, which is known as Tamiflu. And that's for the targets of the viral neuraminidase. Not sure about Xofluza yet. That's a newer drug that was approved in Japan. And so that affects actually the polymerase, which again, obviously if you can't transcribe the viral RNA, you shut down viral propagation. And so there's various influenza antivirals in clinical trials, but none of them target actual NS1. And so this something stood out to me is that, you think if you were trying to find an antiviral, you'd want to attack as many targets as possible. And if you get rid of NS1 in the protein, if you just delete it, the virus cannot replicate because the innate immune response just swarms and takes over and effectively shuts down viral replication. But if you have NS1 activity, which inhibits that innate immune response, you're able to have a productive infection. So what about vaccination? Okay, so obviously this is a hot topic these days. The problem with vaccination, at least for flu, obviously now we see is vaccine hesitancy. And sometimes there's an emergent strain. When one of those recombination events or reassortment events occurs, you have an emergent strain. And then you have about 2 months to manufacture a well-matched vaccine. And this was the case in 2009. So you have a 2-month window as if you have a highly virulent, highly contagious virus. That's plenty of time to do a lot of damage. And so we need sort of a stopgap measure for those either that don't want to take the vaccine, or if there's an emergent strain and then suddenly the vaccines that we have aren't effective. Be interesting to see what they do with the mRNA vaccines, if they're going to apply that to flu.
So it's collected yesterday, right? 11:40. Okay, we need to process today.
So I'm not able to.
I think
I'm
Hall
sorry, Chad, I
has
don't
finished
have
today
those
and he's
privileges.
gone for today.
And so I
Um,
emailed someone to see if
if
they can come
somebody
in and
could
try to take care of
walk-
this.
Walk it
Okay.
to the lab. I
Lee,
can find the time this afternoon to process it. That would be wonderful. Could somebody, or if you couldn't find anybody like walking into the building, just give me a call. Let me give you my cell number. 205-396.
So can you hear me on top of her, or
5160.
should I wait?
we can, it's disruptive.
in
We can hear
Shelby
you, but it's hard to
277.
understand if Lee's
Okay.
talking, obviously.
Yeah, that's the lab.
I'm gonna try to page Laura.
Thank
Okay,
you.
thank you.
Bye.
All right.
Maybe worthwhile seeing, could everybody please mute themselves?
Thanks, Liz.
Yeah, if everyone's listening, if they could please mute themselves, that'd be really helpful. Thank you.
Okay, so we have NS protein 1, or non-structural protein 1, NS1, and it's divided into two subdomains, approximately 230 to 237 amino acids in length, depending on the strain. And it's associated with many aspects of the virus life cycle, including temporal regulation of RNA synthesis, suppression of the innate immune response, strain-dependent pathogenesis, splicing, apoptosis suppression, mRNA translation enhancement, as well as viral particle morphogenesis. And so this was very intriguing to me because, as was mentioned when I did my postdoc, we use a lot of biophysical techniques, and it's very interesting that such a small protein can have that many effects and functions inside the cell. And so these are the two separate domains. These are from a UDORN strain. This is the sort of what we know as the RNA binding domain, and then the effector domain. And so this is, if you get so in between the effector domain and RNA binding domain is a small sort of unstructured region, about 7 amino acids. And so they form independent folding units, and RNA binding domain is folded into a dimer, and the effector domain is actually a monomer. And so if you just look, so today's talk will just concern the effector domain. But even if you look at the effector domain, you have a small protein, and it can interact with, say, PKR, which blocks the antiviral pathway. It has a PDZ domain for some strains. It interferes with CPSF30 function, which limits IFN production by essentially sequestering all the mRNAs to the nucleus so they can't be translated, important alpha, et cetera, et cetera. And so that's what really kind of started driving the question for me, because, such a small protein, is this going on in all strains of viruses? Or is it like sort of like you have some base model where, in a car, for example, and you have, the car can do the PKR and PDZ and CPSF30, but if you include the enhanced package, then you get P85 beta, P15, EIF4GI, and then you can see where I'm going with this is that if you have various different interactions with different strains, perhaps that would give us some sort of information in terms of the virulence potential for the virus. And if you can have sort of a hierarchy of, if you get a combination of PAB, P1, P2, and CPSF30 and CRKL, then that's really going to be an infectious or highly pathogenic strain. And so if we can pinpoint how these structural elements interact with one another, then you can actually inform surveillance efforts for predictive in terms of how bad a flu strain or flu season it might be. So there are some novel inhibitors of, so for example, NS1, this was designed, so it's not in clinical trials, but it was designed. And so it was shown to inhibit replication, is indicative of inhibition of multi-cycles, but not a single replication cycle. And the mechanism of action was currently, when they published this in 2011, not known. These compounds are referred to as A9 and A22. A9 was sort of the initial compound. A22 was much more of the developed compound that was more effective. And so when I first started looking at NS1s, I like to play on Thymol and just sort of see what I can find. And one of the interactions, which was with CPSF30, particularly the F2, F3 subunit of CPSF30, bound to NS1, there was a crystal structure. And when I saw this, I was like, that looks familiar. And then I remembered that these two phenyl rings represented the two tyrosines or phenyl rings on the actual binding, our CPSF30 cellular component. And so I was like, well, I wonder if that's the mechanism, because once again, all they knew is that they had designed it using a yeast-based screen against NS1, but they had not confirmed that it was targeting NS1. And so we actually did some in silico binding, and we found that, yes, indeed, it did bind, at least computationally, to the NS1 effector domain inside the F2, F3 or CPSF30 binding pocket. So that was really cool. But just like anything else with in silico modeling, you really want to confirm this using sort of a bench technique. And I am an NMR spectroscopist. So we wanted to use a term called a technique called chemical shift perturbation. And so to just kind of put it in brief, you first isotopically label your protein, and that gives us an HSQC, which stands for heteronuclear single quantum coherent spectra. Don't worry about that. It's essentially a fingerprint for the protein. Each one of these sort of dots represents an amide bond, or the cross correlation peak for an amide bond. And so you can imagine you have for every amide bond, you have a dot. My kids always make fun of me because I'm looking at the screen, looking at dots, and they don't understand, why am I, do I look at dots for a living? So that was, pretty funny. But so then what you do is, so if you don't isotopically label the protein, then the protein you express is actually invisible NMR due to some quantum physics or quantum chemistry, and I won't go into that. But so for example, the isotopically labeled N15 shows up, N14, which is the most common isotope, doesn't. And so what you can do is you can put in a ligand of any choosing, so for a small molecule or stuff, and since it's invisible to the NMR spectra, you don't see the actual compound in the spectra, at least for this type of experiment, but you see its effects. And each one of these cross peaks is extremely sensitive to the local chemical environment. So if you have a binding event and you alter the local environment, then that peak will shift. Therefore, that's chemical shift perturbation. So what you have, the native chemical shifts unbound, you put your ligand in this case, it was a small molecule, and you see if there's any shifts. And you overlay the spectra, and then you can tell if it's actually interacting. And the cool thing about NMR, okay, so I'm sorry, I'm getting ahead of myself. So these are two of the spectra for, this is for the RNA binding domain, and this is for the effector domain. And so you can see there should be one peak for every amino acid, except prolines, because they don't have an amide bond, and some side chains, because some side chains do have amide bonds. And so one thing you can see from this is kind of like a fingerprint for the protein. One thing that you can determine automatically is that if you have the correct number of peaks, all the amino acids minus the prolines plus the side chains, then if you have all peaks accounted for, then you can say, okay, that's a single conformation. Because if they had two conformations, then each one of those would be experiencing different chemical environments. So you'd have more peaks than you would realize. If the protein was not folded, then the chemical environment that is experienced by an unfolded protein is very similar at all points, because it's just sort of like a wet piece of spaghetti floating around in solution. And so therefore, all the chemical shifts are along the same sort of section. And then you would see basically it'd be from here to about here, and it would just be one streak. And so what you can tell from these is that these proteins exist in one species or one single state, and they're well folded. And so the cool thing about NMR, at least in my opinion, is then you can come in and assign each one of these cross peaks. So now you not only have you have probes for each one of each amide bond, and those probes are exquisitely sensitive to the chemical environment, and you know what the probe identity is. So for example, this is serine 99, this is glycine 158, et cetera, et cetera. So when we actually do the chemical shift perturbation, not only do we know where it binds, or if it binds, we know exactly where it binds. And so we did the chemical shift perturbation, and so this is for A9. So in black, it's unbound, and it's hard to see in the overall spectra, so I blew up some of the sort of regions where we notice these perturbations. And so then you have A9 in green. And so you can see some of these shifts were occurring, which indicates to us that the small molecule is actually interacting with, say, let's see what, arginine 118, lysine 108, et cetera, et cetera. And so we're very excited to see this because it confirmed the in silico modeling. And so if you actually do, you graph the amount of chemical shift perturbation, then you can actually apply some statistics. And then you can see, okay, well, these shifts are statistically significant. And this was actually a technique that was developed because there's no real one right way to do this. Some people do 2 standard deviations, but we found a method that actually does an iterative process, and it's been successful at finding, I think, 95% of actual positive peaks for interactions. And another thing we can do is, since we assigned those resonances, you can look at their relative chemical shift values, and you can kind of map you actually are able to map the secondary structure. And once again, we're able to map the secondary structure. And so we can say, okay, well, it looks folded because this is essentially the structures of other NS1 proteins or effector domains that we've seen from other strains. So, okay, then we went with A22. I'm not gonna, go into detail. It's basically the same thing, except the more modified compound. And as you can see, we still have these basically the same shifts in the same positions. So now what we wanted to do is we wanted to map these changes to the effector domain. And so we then went ahead and solved the crystal structure for the 1918 effector domain, because that's the strain we're using. Ironically or weirdly, this had never been looked at. We had the sequence for the most deadly influenza virus in history, and we didn't have any of the structures for NS1. So we went ahead and solved that with collaboration with Dr. Todd Green in the Department of Microbiology. And I mean, for the most part, it looks like a typical effector domain fold. There are some differences in loops and things, but the overall fold of the protein was identical to other strains. And so once we map these structures, map the shifts that we saw. So for example, you remember I said that each one of the shifts, you can actually know which amino acid is being perturbed, therefore where the binding site is. And we're able to compare that to CPS of 30, because that was our hypothesis that the small molecule mechanism of action is to basically stop that interaction from occurring, which is important to the viral life cycle. So in yellow, these are the shifts that were determined from, or the interaction interface that was determined from the crystal structure. And so when we compared, say, yellow is the crystal structure site map, we had our red is the amino acids that lit up for us in terms of our chemical shift perturbation, and orange is ones that were common to both the crystal structure and the NMR data. And as you can see, they're highly overlapped. Then we tried 2013 with A22, because once again, if you have an antiviral and it's only good for one particular strain, then the next year the antiviral is no good. So we wanted to see the broad specificity for this potential broad specificity potential for this particular antiviral. And once again, we saw that even from a relatively modern strain, 2013 avian influenza, we saw the same path or binding interface light up. So we were able to conclude. So we actually published this in the Journal of Biological Chemistry. My computer froze. Let's see. It's one of the benefits of Zoom, or not the benefit, but okay. My apologies. Computer's frozen.
We're getting to experience all the technical difficulties.
Well,
the good news is
there we go.
Okay.
Cool. Okay. So the we actually published in JBC. We actually got the cover that for that month. This is in the effector domain with all of the different poses that we saw in silico. And so the conclusion we draw is that you have A9 and A22 are novel antiviral compounds. that have chemical shifts that bound to both compounds bind to an affected domain. We mapped the perturbations, and then we found that overlap with CPSF30, and it's actually potentially has broad spectrum activity. So, okay, so that sort of finishes up that part of the talk, and it froze again. Let's see. Am I still sharing my screen, or did that go away?
Yeah, the slide is the conclusion slide for
Yeah, okay.
JBC.
Perfect. Okay, cool. All right, we good? All right.
That's where, yeah, that's where you were before, though. I don't know if it's advancing or not.
Yeah. Okay. So that sort of wraps up that part of the talk. And if anybody has any questions, I'll be happy to answer them. But we can move on to the other sort of what the more the applicable to the title was. And sort of now that we finished that, I just wanted to show how important the development of antivirals were, plus, you know, NS1 being an effective target. So once again, you know, just to recap, you know, the affected domain plays many, many roles. And as you can see, it interacts with many, many proteins. And it's known that the effector domain is, it gets shuttled between the cytoplasm and the nucleus. And one of our hypotheses, in addition to some of these being strain dependent, is that maybe there's a localization of sort of correlation in that, you know, obviously if you're in the nucleus and you can't interact with the cytoplasmic protein and vice versa. And so one of our thoughts is that maybe some of these interactions that are more important are dependent on whether the NS1 protein is in the nucleus or cytoplasm. And we did this, we actually wanted to see sort of, we went in and tried 4 different strains of NS1, or the NS1 from 4 different strains. We have 1918, PR8, which is a lab-adapted mouse strain, 2009, which I said previously is not particularly virulent, and 2013. So we have 2 highly virulent strains and 2 not so virulent strains. And when we did this, so this is a confocal microscopy. We ectopically expressed NS1 from multiple strains. And what we saw was pretty fascinating. And so you can see from the cytoplasmic side, you know, you have these like punctate localization sort of centers for 1918, and a little bit more, less, but still pretty punctate in 2013. Whereas PR8 and 2009, the 2 strains are not as virulent, are really sort of more diffuse. And this led us to think, you know, like, well, something's going on, because this is just NS1. It's not like part of the viral, you know, it's not during a natural replication, but something's causing a difference in localization patterns. And so what we did, and these are obviously the 2 highly pathogenic strains. And so we actually said, okay, so now that we know about, we have this weird phenotype, or this weird result from confocal microscopy, how does NS1 modulate or regulate its own intracellular localization? And what we found is there's not a lot known, but what we do know is that you have this export signal, 138 through 147, which is in green, and this inhibitory signal, 148 to 161. And we do know that intracellular localization is strain dependent and correlates with virulence. And so that was interesting. Okay, so, and it's controlled by a latent nuclear export signal that is hypothesized to be activated by dynamic movement of the adjacent residues that mask the signal. This was very interesting because you know, you have the signal, and then you have the inhibitory signal right next to it. So how is that interfering with, you know, the export? And, you know, what is going on there? And this is a very highly conserved region. And so that put another sort of caveat in there, is that this region of inhibitory signal and nuclear export signal is really highly conserved. Yet you have these differences that occur in naturally occurring mutations or naturally occurring strains. What is going on? And so, like I said earlier, this might be essential for NS1's ability to perform multiple functions. For example, in the nucleus, it can interact with CPSF30. In the cytoplasm, it'll interact with RIG-I, PKR, and PI3K. So what we did is we did basically just, we wanted to see what were the differences in amino acids, or if there are any mutations that were common to high path strains versus low path strains. And we found one in particular. It's aspartic acid 171. It's found in 1918 and 2013. Both are highly virulent, obviously. And in PR8, it was an alanine. And in 2009, it was a tyrosine. And so we started really focusing in on this particular amino acid. Now, so to give you a context of where this is, once again, the green is the export signal, the red is the inhibitory signal, and 171 is not found in either one of those. But what we did find was that 171, when it's an aspartic acid, coordinates this water or electrostatic interaction with arginine 140. And so this was our structure of the wild type effector domain from the other paper. And then we looked at, okay, what is the conservation of 140 and 171? And like I was saying earlier, is that 140 is really conserved. I mean, it's almost 100% arginine. But 171 is 50% lysolucine, 30% tyrosine, et cetera, and aspartic acid is 2.2%, which is, I understand, maybe statistically it's not, so you'd have to do some statistics, but this was really striking to us that, you have one of the fewest represented amino acids in aspartic acid, and yet you have two viral strains that are about 100 years apart, and they both encode the least amount, or the least represented amino acid. So we then we wanted to say, okay, well, what happened? What is going on with the structure? Is it any structural perturbations? Because, like I was saying, obviously the inhibitory sequence and the export sequence are right next to each other. So something has to be going on that releases the inhibitory sequence. So maybe it's a structural change. But when we solved the structure, it was interesting in that the structures were nearly identical.
Chad, can I interrupt just for a moment? Yeah. It still shows the conclusions JBC slide on our screens from probably a few slides ago. You might stop sharing and then just retry again. And if that doesn't work, just keep going.
Okay, so all right, so,
Are you on a different slide?
Yes.
Okay, we still see the conclusions JBC slide.
Crap. Okay.
I'm a little technically challenged, if you can maybe stop and then just restart it, it might work.
Yeah, I'll do that right now.
I'm sorry to interrupt. Thank you.
No, that's there we go. Is that better?
So that shows us the current slide and the next slide, which is better than what we had.
Oh, okay. The current slide and the next slide. Oh.
So we kind of see the presenter screen, I think.
Oh, okay. That's weird.
But for the sake of time, it might just work.
Yeah. Oh, let me see if I can.
I mean, it looked okay before, but we can still see the next slide just for what it's worth.
My apologies. Seems like we're getting hit with every Zoom.
It's okay. We're patient.
How's that?
So that looks great right there. Yep.
Okay. All right. So, and this was the slide I was on. All right. So I'm going to just blow through what I just talked about just to kind of give a complex, or if you can, I'll just hit the highlights. So effector domain, obviously a lot of proteins interact. Some are in the cytoplasm, some in the nucleus that could attribute the multifunctionality. So this is the intracellular localization I was telling you about. 1918, 2013 are really highly virulent. 2009, PR8, not so much. This, the green is the NS1. Oh, wait, hold on. The green is the NS1, and you can see how it's punctate in both 2013 and 1918, but very diffuse in PR8 in 2009. So that led us to wonder what's going on. Here's the structure of the NS1 effector domain from our previous paper. In green is the export signal. In red is the inhibitory signal. It's weird that they're very close together. It's not really known how they mechanistically function in terms of inhibition and activation. So then we found an aspartic acid at position 171. The other proteins include alanine and tyrosine respectively for PR8 and 2009. So when we looked at the structure, you can see here, so for aspartic acid, 171 and arginine 140, you actually have this coordinating water plus this other potential electrostatic interaction. And so that's interesting to us because, like I said, you know, our aspartic acid is the least represented amino acid at that position, yet it's common in both the two of the most highly virulent strains of influenza. So then, okay, so great, you know, whereas the arginine is 100% conserved. So now we wanted to see, okay, what if we saw the structure of the aspartic acid mutated to alanine, which, okay, and here. And so what you can see is that, so in gold is alanine 171, that's the mutation, and D171 is the actual aspartic acid that's still there. And you can see how this coordinated water occurs when there's an arginine 140, which is consistent throughout all strains. Whereas when we have the alanine 171, all of a sudden this arginine flips back out. There's no more coordinated water. And so you're getting rid of this electrostatic interaction, which one hypothesis that we have is that that allows more of a dynamic process to happen, because this might serve as sort of like a clothespin or a clamp to allow, you know, certain secondary structures to move more freely, therefore exposing the export signal and or inhibitory signal. So, okay, so just rest assured, everybody, I did not want to recreate mutant 1918 Spanish influenza viruses, so we switched to PR8. This is the lab adapted strain. It's a good model for NS1 activity. I mean, at least NS1 for the effector domain, because it has 97% identity. The only differences are 106, 103, and obviously the A171D, like we were talking about. So on the left are amino acids that are found in PR8, and right are their corresponding in 1918. And so, so if we want to do any viral work, we had to actually switch strains. But because the effector domains are highly conserved, we thought, you know, and we're only looking at how this one amino acid affects virulence and things like that. So, you know, it's a pretty good system without having to make one of the most deadly viruses known to man. Okay, so then you have the consequences of mutating position of 171. Now, typically NS1 and NS2, which is now known as NEP, just to confuse things more, are made from the same NS segment. And that's due to a splicing event that leads to NAP synthesis. So if you make a mutation in the NS segment that you need for NS1, you inadvertently make one for NS2 as well. And so we were very aware of this. And so there's no way to mutate 171 to aspartic acid without actually make a corresponding mutation in NEP. Lucky for us, though, is that position 14 was shown not to have any effects on function. So, so we can look at our potential mutation in the effector domain without worrying about it altering NEP function. And so when we did the transfection, so we transfected NS1, both the mutant and the wild type, into 293 cells. And what we notice is that the mutation, A171D, caused enhanced nuclear retention of NS1. And you can see here for the mutation, most of the NS1 is in the nucleus, whereas it's in the cytoplasm for the wild type, PR8 strain. And so that was like, okay, well, so far so good. We have a phenotypic difference that sort of goes along and supports our hypothesis that this particular amino acid, while being outside of the signals, inhibitory signal and export signal, actually affects function. So the next part, we wanted to generate novel influenza viruses that harbor the mutation. Once again, we're going to use PR8. So we have what's called a reverse genetic system. And essentially, you have 8 plasmids, each encoding for the H segments. And you can transfect all of these plasmids into a cell, and the virus gets made. And so now you have a recombinant PR8 virus. And so what you do is you just take this NS1 segment, or NS segment, sorry, make the single mutation at position 171, do the same process over, and then you have a recombinant PR8 wild type virus and a recombinant PR8 mutated virus, A171-D. So, okay, so when we looked at the replication kinetics of both the wild type and the mutant, the wild type is going to be in black, the mutant is going to be in red for most of these slides. And so there's an enhanced replication in multiple distinct cell lines. We used A549, which is an adenocarcinoma lung cell. We use normalized human bronchial epithelial cells, which are as a primary cell line. And we have MTEC cells, which are mirroring mouse cells. And so it's interesting to us is that the virus titers appear to converge only in mirroring cells. So that suggests it could be some sort of species-specific thing happening. And so then we did a time course of nuclear retention of NS1. And so it's kind of bizarre, but when we looked at an MOI of 2, so that means we infected with a high number of virus particles. And so 10-hour post-infection, we saw that for the wild type is predominantly in the cytoplasm, as you can see here, whereas the mutant was represented in the nucleus as well, this overlay. And then 12 hours, they look much more similar. And so A171D is predominantly cytoplasm, and the wild type is also predominantly cytoplasm. So it's interesting to see that this difference in phenotype only over 2 hours. So just to be sure to note, it is not clear at all what causes export and what, besides the inhibitory sequence, there's got to be another protein in play that masks the inhibitory sequence and that's then released, or masks, something along those lines. And so that's one thing we currently are looking at. Not only that, but it is not known which exportin takes NS1 out of the nucleus. So there's a lot of still things to figure out here. And that's what we're doing. So I said, okay, what about type 1 interferons? So we looked at IFN beta, IFN lambda, IFN beta in terms of, different levels of, or different time points. And so we looked at IFN beta, for example, and you can see the A171D mutation does in fact induce higher levels of type 1 interferons, at least from the RNA perspective, as well as lambda. We kind of went a little bit further out here, 24 hours post-infection, still higher than wild type. And an MX1 is a reporter for type 1 interferons. And once again, we saw the same effect. Take home, the mutant induces higher levels of type 1 interferon mRNA. And then we decided, okay, well, let's see how pathogenicity-wise in mice. So we looked at the survival, and the A171D mutation, I think only one of the mice died, whereas was just wild type, we had 40%, only 40% live. And as you can see, the weight loss, interestingly, when mice are infected, temperature goes down with influenza, and also the clinical scores that are more of a subjective, how's this mouse look, and you just kind of have to rely on certain ways the mouse is behaving to have a certain clinical score, but everything matches up. I mean, essentially, the mutation is less virulent in C57BL6 black mice. And so now we wanted to, also look at some of the titers in the mouse lungs to see if we can come up with any sort of theory as to what's going on in terms of, is it IFN beta, or is it a innate immune response, or is it just replicating better, et cetera, et cetera. So interesting, PR8 wild type replicates higher titer in mouse lungs. And so you can see, once again, black is wild type, red is mutant. And in day 2, day 4, and day 7, you have higher titers of virus. Once again, it kind of makes sense, more virus, more death in the mouse. And so, okay. And interestingly is that we then performed RT-PCR with mouse lung homogenates and found the wild type induces higher levels of interferon, type 1 interferon, beta, in terms of particular different days. And that was interesting to us because usually there's an inverse correlation between the ability for a virus to replicate and the production of interferons, right? Usually it's more interferons, less virus, not more virus, more interferons. And so that's very puzzling to us at the moment. And so just the conclusions that we have is that the mutating position at 171 abolishes this electrostatic interaction. We're currently working on trying to figure out if this represents a dynamic change, because that's what I did in my postdoc. Sometimes when you do X-ray structures, the structure looks exactly the same, but the motions are different. So we're using NMR to figure that out. It alters localization, intracellular localization of NS1. The mutation replicates more efficiently, induces expression of mRNA to a higher degree than wild type and human direct sidelines. The mutation is less pathogenic in mice when compared to wild type, and in mice, wild type replicates more efficiently, induces the expression to a higher degree than the mutant. And so that sort of, so this is sort of the future questions that we're looking. Traditionally, mutants perturbing NS1 function result in a negative correlation between IFN and replication, whereas that is not the case here. And also, we have different levels of understanding in terms of what's happening in species, because we have different results in mice and in human cells as well. And so that's another sort of aspect of the project that we're looking into. And like I said before, here we have just, the structure looks basically identical. What is the difference between these two, proteins that's causing this level of change in the intracellular localization, and then hence affecting pathogenicity, affecting interferon production, et cetera, et cetera, from a single point mutation that's actually naturally occurring. And so is it a species thing? Is it a dynamic thing? And so that's what we're looking at. So with that, I just wanted to say thank you for everyone. This was taken a while back, and the person that did most of this work is Alex Kleinpeter. He's now at NIH doing a postdoc. Alex Jerica is a postdoc at Georgia State. Andrew is now in private industry. And so they have, all three have graduated. So now we have sort of PT Lab 2.0. There's Morgan, Andrew, Meredith, and Lauren. And Morgan is the one that's actually starting to do knockdown assays to determine the exportance and things like that. And so, sort of following up on these questions. So with that, I'll just take any questions.
Thanks a lot, Dr. Petit, and apologize for some of those disruptions from earlier.
That's all right.
If anybody has any questions, feel free to jump in.
Chad, it's Kevin Harrod. Very nice talk. So have you thought about looking into some of the more early events in the mouse model to understand why this mutation leads to, increased survival in the mice? Yes.
So that's something. Actually, Kevin's one of our collaborators. I forgot to mention his name specifically, but he's on the last slide. So yes, I mean, that's a very good question. And we think that's where all of the sort of activities happening, because we don't have actual specific quantifiable data, but we've noticed that if you, even if you push the sort of advent of the innate, sort of like the innate immune response hands it over to the adaptive response, in theory. We found that, at least, anecdotally, or you're looking at the mice, there's like a day, if there's a day lag in the innate immune response, then that mice dies. Whereas if the innate immune response actually holds out for, say, 4 days or so, and is able to effectively hand it over to the adaptive immune system, even if that mouse is really, really sick, it lives. And so that's what we're trying to figure out in terms of the interferon. Is that correlated with intracellular localization? What proteins are in play? especially if since the intracellular localization appears to correlate with virulence, why is that? Is that interaction? I would imagine that interaction is very significant, whatever protein's causing that to happen. So, but yes, I mean, sort of the problem with this particular, the problem with going from, essentially structure all the way to virulence is there's a lot of stops and off ramps in the middle. And it's just a matter of figuring out which one to take to figure out, answer the question. So.
Chad, I have a question. First, thank you for not going into the quantum physics of NMR. That was very much appreciated. But it sort of follows on from what you just, from Kevin's question and your answer about the perspectives or the considerations from therapeutics in terms of, you've got a pretty small window, at least from the mouse studies, right?
Right.
So how does this sort of translate out in your mind? I mean, is this going to be a feasible target therapeutically when you think about the therapeutic window?
Yeah. So the therapeutic window is small, right? But that's based on our finding that the mutation doesn't prevent export, it only delays it, right? And so our hypothesis is that if you could prevent it with, say, like a small molecule, or you can prevent import, then that would effectively shut down the virus and give us much more of a time window, because right now it's preventing it from, it's delaying it from getting out. It looks about 2 hours. And so if we're able to modulate and effectively allow that window, to be greater, maybe with additional mutation or a small molecule or a protein or something, then that would much more be applicable to a therapeutic of some type, some type.
Oh, I see. So you wouldn't have to. I was thinking more that this would be more of a personalized or precision approach, but you're really talking about allowing it to become a precision approach, right?
Yeah. So essentially, just trying to figure out what if you understand the mechanism of NS1 export, then that's a critical step in the virus, and we're showing that export temporally affects virulence. And so if you can modulate export in a certain way, then you could actually, stop the viral life cycle essentially and allow that's another issue with NS1 as a therapeutic target, because, for example, so you have a polymerase inhibitor. You can give the polymerase inhibitor, you can test it, and it essentially shuts down the virus, right, mechanistically. NS1, on the other hand, if you target NS1, it doesn't really do anything to the virus life cycle itself. What it does is it stops the virus from interfering with your body's natural ability to get rid of the virus. And so it's almost like stopping the virus from blocking your own immune system. And so that's another reason why I think hasn't been as attractive, because it's hard to do that, because there's no, if you have a molecule or a compound, you can just throw it in the cell or the dish, see if the virus can replicate, and then if it can't, good. Then you worry about bioavailability and stuff like that. Whereas this is like, okay, well, how do you test NS1 function and that type of thing? And so therefore, because we know if there's no NS1, the virus gets cleared easily. And so this would be allowed more of like allowing your immune system to sort of an immunotherapy almost, without actually modulating your immune system, just allowing it to do what it naturally does. And also could be used as part of a combination therapy to, if you target multiple viral proteins, that could also be very beneficial when inhibit escape mutants as well.
Thanks.
Thank you. Any other questions?
Thanks again, Dr. Petit, for a very excellent talk.
Thank you for the invitation.
Yes, and I want to thank everyone for being here this afternoon. Next week, Dr. Benveniste is doing a talk on critical contribution of neutrophils to autoimmune neuroinflammation. So tune in next week. Everyone have a good Thursday. And thanks again, Dr. Petit.
Thank you.